also address the lack of validated biomarkers. And that's the second big thing that we've tried to discuss here at the podcast. And I think through the work that Mazen, Nureddin, Naim Al-Khoury, Rowe, Lumba, and also Lina L have been doing around imaging biomarkers, MR, the field has just evolved, and we're clearly, it feels like, taking big steps forward.
It's interesting you should mention that, because shifting away from this episode to some of the other things that we're going to be replaying for people this week, on Saturday we're going to be looking at 3 conversations, 3 little 15, 18-minute snippets from episodes that were extremely well downloaded. The biggest one, of course, was the one with you and Stephen summarizing what you heard from Scott Friedman and Lars Johansson at the Paris NASH. But one of the other 2 is specifically about that. It's what was the takeaway from AASLD 2021 on the issue of the need to improve biomarkers and diagnostics in general, but really non-histopathological, both MRE and the liquid biomarkers. And yeah, I think that's been another major theme in the field over the last 2 years, right?
Yeah, absolutely. If you just revisit the last ILC, it's not just that we're in the position to actually define those diagnostic biomarkers, but we're starting to discuss prognostic biomarkers with ELF that has been received approval by the FDA in certain populations, histology at baseline, or the whole data sets that we've discussed with the work that's been done by Hannes Hagström's group, or recently presented by Quentin Ansi with regards to outcomes in FIP4 or high categories, the differentiation between liver outcomes and cardiovascular outcomes. That feels like light years away from what we were thinking and knowing in 2020, because we're much more focused on outcomes, the role of surrogates, which patient population should be included in clinical trials. And if I think back up to our conversations, we had a lot of these aspects were discussed here and in Surfing NASH, and that's what I value the podcast for. It's really state-of-the-art science that's important to the field that's being recapitulated here.
Yeah, I agree, and frankly, it's a lot of fun. When I decided to do this week's episode, I didn't realize how much fun I was going to have listening to old episodes and saying, gee, people have really, changed tunes, and thinking about moments in papers that did that and possibly ways that the podcast helped to shape it. This was one in season 2, episode 57.1, which was part of the takeaway from AASLD. We went back to Stephen's phrase, which is about putting a big fat dent in fatty liver disease. And the specific question was, okay, what happened?
I was going to say, which one of Stephen's phrases?
Yeah, one of. In a whole different context last night, I was listening to somebody talking about American politics, and they used the phrase, is the juice worth the squeeze? And of course, that brought me back to Stephen also. But the specific question for this conversation was, what did we hear last week that did the best job of putting a big fat dent in fatty liver disease? And most of the focus was on that. It was on using NITs and MRE as a way to build a bridge past, individual biopsy reads, not even AI biopsy reads, but basically histopathology reads, to an NIT future. And the steps that in the fall of 2021 we could see ourselves taking in that direction. Yeah, when you think about how the world is different than it was 3 years ago, 2 years ago when we started the podcast, to me that's probably the biggest one, is how people are thinking about what to do in clinical trials and what we know about the underlying disease that we didn't know or didn't really put together the same way then.
I'm fully aligned, Roger.
My icebreaker question is actually a bit of a tribute to Stephen, except the one to Manal, which is actually a tribute to Manal. There are 2 really compelling talks I had in my life that got me involved in fatty liver disease, and Manal was the second, as she knows. Stephen was the first. And one of the things that Stephen said during his was that his mission in doing all this was, and I'm quoting now, to put a big fat dent in fatty liver disease. So the question I want to ask the rest of you, and Manal, I'm going to ask you this too, but as part of a broader question or a different question, is, and Stephen's here.
Hey, I'm here. Sorry, guys. Just had some clinical things going on.
Stephen, your timing is off by a minute and a half. Here's today's icebreaker. I was saying that I got involved in NASH in the first place as much as anything else because of a conversation I had with you and one with Manal. And for the icebreaker, when we first talked, you told me your mission was to put a big fat dent in fatty liver disease. I want everybody, except Manal, who will tell us that in the context of a broader question. Where do you think the biggest dent got hit on fatty liver disease this week? Brave one, go first.
Louise Campbell.
I actually think it's probably the genomics sessions, although I didn't cover any of those. There were quite a few interesting things from what I could see looking at phenotyping the people who aren't going to respond to some of the drugs, different diets, things like that. And I think the more we drive down into that precision medicine, the more we get into areas that could really change the landscape. Did these drugs fail? Or if we looked at some of those studies that failed with new techniques that we've got, could we pick out the patients? And therefore, were the drugs more successful in the patients from the right phenotypes? That would be the sort of thing that I took from this meeting that I hadn't seen previously.
It's a great answer, Louise. Thanks. Okay, now whoever goes next can't repeat that.
Jörn Schattenberg.
All right, I'll go next. So for me, the big dent it really comes in from the fact that we're talking with different physicians beyond hepatologists to how to identify these patients, to make a difference for these patients, to actually find these patients. Albeit no approved therapy is available today yet. We're discussing these strategies, how to identify them in different settings, in different healthcare systems. And this is a big step towards the realization that the disease is important, it's real, it's critical for patients. There was a special prize given to Dana Cryer for her advocacy. This is really the momentum that we've reached more people than just the experts with this critical disease.
Thanks, Jörn. We're reserving Manal for last, so Ian and Stephen, go ahead.
Ian Rowe.
Well, I'm going to go next. I'm ready to follow on from what Jörn said, and that is that I thought that this was a meeting that was a bit about research, but a lot of it was about practice and about how we might implement treatments that are not drug treatments now for the benefit of patients. So there was a real focus that came through the meeting on diet and exercise interventions. That's to the benefit of our patients today. It reflects a wider recognition that we need to do something now, and we can't afford to wait for drugs that will be coming down the line.
Okay, thanks, Ian. That's great. Stephen?
Stephen Harrison.
As I reflected on the meeting and reflected on, first of all the great talks that were given, and many of those by you guys. And by the way, before I go on, I'll say great job, Manal, on your wrap up. I thought that was very well done. And trying to go through his 475 different posters and whittle down what was the most important thing to drive home was very, very challenging. So congratulations on that and your two children for their help. But I think for me, the big fat dent really, I see the tide turning on some of our non-invasive tests. And in 2022, we will have some significant data that will hopefully support the initial data that's come out on some of these NITs, particularly as it pertains to the context of use of diagnosing the at-risk NASH patient and monitoring efficacy of drug. Still think we got a ways to go on outcomes, which ultimately is the holy grail, but the groundwork has been laid for us to mine that data, and we'll hear that in 2022. And I believe that groundswell has begun at AASLD 2021. There's a lot of things I could say that left a dent. To me, that in my mind was the biggest dent.
Thank you, Stephen. So I'm going to steal about half of Louise's and half of yours, and I'm going to say that the big dent that I felt was an increasing acceptance of the complexity of the disease. I talk about this from time to time, but in 1969, Nixon declared war on cancer as if there was a single thing called cancer. And one of the things that struck me at NASH 2019 when I first came here was the degree to which people were talking about as if there was a thing called NASH. And what these discussions have been about in a whole bunch of directions is understanding far more richly what the disease is, and then how it might differ from patient to patient and how to understand that and how to treat it. All that richness, Stephen, has the effect of walking us away from biopsy, because biopsy is a really blunt instrument, and away from looking for one drug that's the holy grail, because A, that hasn't worked for us so far, but B, the more we know about the disease, the more we know that that's probably not the right thing to do. So I just thought the whole thing got a lot richer.
Just one caveat. Biopsy is actually a very sharp instrument, but the results are very blunt.
Can we call it a sharp instrument and a blunt analytical tool? Will that cover the landscape? Very good, Stephen. So now I have two questions for you. The first one is, and I'm not the only one who's asked this, how many med school acceptances did your kids get last night, and how many came with scholarships?
We haven't applied yet, and I don't know. We'll hope the scholarships will come in later.
In Division I athletics, a 14-year-old kid with talent can get a scholarship offer simply based on that. And I think if there's such a thing in med school, your kids certainly put themselves right at the front of the pack last night.
Manal Abdelmalek.
It wasn't a hard task. It was highlight every time you see NAFLD, NASH, steatohepatitis, or fatty liver, and then give me those abstracts.
The real question, though, is, Manal, could they pronounce steatohepatitis?
We worked on that. They could after they eventually did.
Okay, so here's the second question, and this is my version of Big Dent for you. Without quite putting it that way, the process of getting the 475 articles highlighted by your kids or posted into that 30-minute talk, to some degree has to be about the question, where was the dent, right? It's what matters. But some of what matters is how much do I have to get in, and part of some of what matters is, okay, what mattered most? So how did you address that?
Well, as I was going through those abstracts, particularly as it pertained to the therapeutic landscape, and I commend Stephen and everyone else who really took the podium on many, many different fronts in that regard, particularly with early phase 2A and 2B trials. What really put a dent in it for me and how I framed the talk the way I did is looking at the early phase trials that show early evidence of efficacy. It was somewhat surreal for me when I got to pivotal studies that ultimately failed. I chose, and it was a very hard decision for me to make, to present the top-line data for the Alpine trial, Falcon 1 and 2, the Tandem study, because for all practical purposes, the proof of concept, the early phase trials, the mechanistic principles behind these compounds, we should have seen a signal. We should have seen some outcome results, and it was somewhat surreal that they failed on the primary outcome of how we've defined this disease, a histologic definition, but yet so many other surrogate markers of disease activity dropped across all these trials that, quote, failed. I put that front and center because I think it framed the rest of my talk, which was really to put into context that this meeting was so robust, and I hope all the regulatory agencies were listening, and brought forward the non-invasive approaches to not only diagnosing, but really risk stratifying and, more importantly, defining outcomes. And whether those platforms are going to be AI-based platforms for better interpretation of histology, and it was really amazing to see, and I can't recall the first author, it might have been Moises, for which the light microscopy was deemed to be unchanged, but AI platforms and pathology was able to stratify that unchanged sector into a progressor or a non-progressor. The pathologist was really good and concordant with AI platforms when there was clear progression or regression, but every time they called it no change, their AI picked up subtleties that weren't otherwise seen. And now we have non-invasive markers that are leading us towards removal of the biopsy altogether, to predicting a clinically meaningful outcome, whether that be cardiovascular or hepatocellular carcinoma or decompensation or death even. And we need to move ourselves away from, as Stephen so sharply said, the sharp endpoint, which is so blunt and nondescript. And as I was entangled in all these abstracts, I wanted to deliver a message. Look how far we've come, but look how much we are failing, and we need to reframe our approach. We need to rethink about how we're defining our endpoints. We need to look outside and beyond the biopsy to more sensitive surrogates to define such endpoints, or we're going to potentially set ourselves up for more failures unless we learn from past history and capitalize on the very robust platforms, technologies, and innovative science that we saw brought forward here when it came to genomics, proteomics, and modeling approaches to optimizing the area under the receiver operator curve for performance of imaging coupled with score or surrogate biomarkers as predictors of outcomes.
Manal, what's interesting is that Stephen made a similar kind of comment last night, except I don't think it presupposed a solution. At least not the way he said it last night, right? We're getting to the precipice. Something has to give. I hope it's the biopsy. Can any of you see a realistic scenario under which it isn't the biopsy that gives, the scenario under which that maintains primacy for another few years, leaves us basically struggling as we've been? Or do you think at this point it's virtually preordained, even if you can't tell the path, that's where it's going to go?
The point that Manal raised is at the heart of drug development in our field here. We need something to have an accelerated approval to avoid having a drug in patients on trial for 5 or 6 years to measure those outpoints. And we've discussed this numerous times on the podcast, and I think Manal highlighted it with her NASH debrief here, that albeit there is effects that we can measure that are biological, plausible, and pathophysiologically relevant to the patient, we're not meeting the barrier the regulators have set to get a drug approved. Now, what would be the best solution? I think we're struggling with that, but we're seeing good advances, and that's what Stephen mentioned with his comment on really improvement in NITs and combinations of these. So I think it's a dialogue. And something I was going to say that I also noticed at this meeting is that the regulators were right there, and there's a number of these think tanks where both Frank and Ania or Laura Dimmick also showed up and talked and discussed. So I think this is really getting us all together and finding a solution for the best of our patients.
The simplest way, whether or not this is feasible or not, I don't know. The simplest way to get to an NIT would be to capitalize on the work already done by many with MR elastography. So just follow my logic. If we know that a one-stage improvement in fibrosis is linked to improvement in outcome, as recently published in Hepatology, off work that Arun Sanyal was the first author on and presented at last year's meeting of AASLD. If we know that, and we also know that MRE is linked to outcomes, as was presented at this meeting in a late breaker, as was published by Alina Allen and Mazen Nareden already in 2 different separate independent cohorts. Now we have 3 cohorts showing this association. It seems like all we're missing is what magnitude of effect change from MRE is linked to a one-stage improvement in fibrosis. And we have the data today to answer that question. That comes from one of the phase 3 trials that's underway right now, where MRE is tagged to biopsy at baseline and will be tagged to biopsy at the end of treatment. If the premise is that the drug is effective on fibrosis and you're able to show one-stage improvement, what does that number need to be for MRE? And when you've got 1,000 patients where that's done in prospectively, that's pretty powerful data that could be presented to the regulatory authorities to say this change is linked to a one-stage improvement in fibrosis and therefore should count as a surrogate, just like fibrosis improvement of one stage does. Now, that to me would account for reaching a surrogate approvable endpoint. You would still want to follow those patients long-term, but can't that be done now with MR elastography rather than biopsy?
You're assuming that the biopsy is the gold standard then, because we've already seen that there's some poor even agreement on, at times, what a one-stage improvement in fibrosis is across different reviewers. So it gets to the point of validating a surrogate marker against histology when histology in and of itself is a poor surrogate. And I've struggled with this rationale. So if you're validating something that's better than a poor surrogate, then you're only making it poor, as opposed to using it as a standalone, independent predictor of an outcome.
Manal, that'll come. That'll come with time. I'm just thinking in the short term, how do we bridge that to become acceptable?
We would need to do it where we have consensus, probably against artificial platforms for interpretation of the histology, for which we have more optimal sensitivity in fibrosis measures, and use that by which to validate the MRE.
That's a good point. We actually have that data now from HistoIndex. 16.94% improvement in collagen links to a one-stage improvement on an ordinal semi-quantitative scale. That's unpublished data, but that's data that's actually out in the domain and can be spoken about. If you wanted to go that way, again, I think at the end of the day, what we need to do is, as this groundswell builds about the variability that exists with biopsy, and concomitantly and in parallel, the groundswell is building for some of these NITs, is that it's time, and we've talked about this through the Liver Forum, we talked about it through the NASH-SIG, we've talked about it through Summit, we've talked about it through other consortium about we talked about it at NASH-TAG, and we will have a fireside chat with the regulators, with key opinion leaders, with industry representatives at NASH-TAG, where the whole idea about this is to show the data with NITs, show the issue with histopathology, ordinal scale, semi-quantitative interpretation, and then see if we can reach a common ground on what it takes to achieve replacement of histology with an NIT for conditional approval. Because I think we've got the outcome piece in the works. It's just that initial piece that we need a little bit more data on. I get asked all the time, well, what percent change in MRE is linked to improvement? And we can speak to 15% worsening in MRE is linked to worsening of disease, just like we can with FibroScan, and we can with ELF, and we can with ProC3, but we don't really have that magnitude of effect change yet. And the problem is, not enough sponsors are doing NITs in phase 2B linked to histology. We just don't have that data set. And then when we looked at phase 3s, we didn't get it with Regenerate. We didn't get it with Resolve It. We didn't get it with Aurora. We're kind of now falling back to Madrigal and the Maestro NASH trial to say, give me what you got. You got 1,000 patients with MRE and ELF and ProC3 and a combination of these that are paired to histology. And if the drug actually meets its endpoint, now we can begin to do some of these post hoc analyses and add fuel fuel to the fire to go to the regulators and say, okay, we got it. Now let's at least accept it as a surrogate. We'll still need to do the long-term outcome measure. We reduce the variability of what these endpoints are right now that are potentially inadvertently killing drug development, as you mentioned last night with aldafermin. I mean, we all think that drug works, but we get one read with 1 milligram. It's the way that statistical analysis was done with MCP mod statistics. You had to look for a dose-response relationship in fibrosis. It wasn't there, so the study was killed.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please send an email to questions@surfingnash.com. We'll be back next week with a new episode of Surfing the NASH Tsunami. Until then, stay safe, surf on, and we'll see you on the podcast. Bye-bye now.